here and talk to you about the study that we have here on premature aging and XP and CS. And really it's any genome instability disorder, so it encompasses a lot of diseases in this study. I wanted to first highlight our pediatric dermatology team. Christina Bull, who will be talking later on today, is part of that, and then Dr. Laura Neidernhofer's lab, and which I am a part of, has really been the research side of this. So we have the clinical side and the research side. It's really a big collaboration between the two groups. So this study includes, it's a registry, so basically it's a bank of data. So a lot of what we've been talking about, so Peter has the natural history one. Ours is kind of more focused on the effects, sun effects, because we have the dermatology group that's very into this, and so they're interested in understanding the skin effects of CS and other genome instability disorders primarily, but also how we can help improve that too. So a lot of the questions might be a little bit focused on sun exposure and skin effects that the patients might have effects in, but then also it really helps with rare diseases because it's pulling all this information from all these different patients from across the country that may not always talk about some of these things. And you only need to contribute once. This isn't a history one like Peter's. This is more of a snapshot, but it is still helpful too because all of the subjects and everyone that participates are at different ages. So in essence, you can kind of get a full picture from a larger cohort. So some of the questions we're looking to really address, and a lot of them are health ones. Again, like how do we prevent skin cancer? Now again, that's also most focused on XP and Fanconi anemia that are part of this study. Is there a way to prevent other cancers or predict who will develop them? So a lot of that's dependent on the DNA repair that we measure. A lot of times DNA repair defects lead to cancer predisposition. And also, how do we slow down that accelerated aging we see in all of these genome instability disorders? So some of the biochemical and cellular research aspects, it's again focused on the DNA repair, how to measure it and see differences between patients, and also and with different gene mutations is all really relevant for us to characterize and understand. And looking at again how these cells how are they aging faster? What does that look like at a cell level? And then do we see the same thing? So we also have participation from parents and siblings in this study, and we can then look at their DNA repair or other things and have that as a comparison to see what is happening that's different in these patients. And then also look at what kind of changes do we see are happening over time, and are those things we could measure as an outcome in a clinical trial? Say we gave a drug or a therapy that we think is going to improve or reduce the premature aging or the acceleration of that aging. How are we going to measure that, and how are we going to capture that biochemically, basically? So looking for measurements that we can do in blood to really see, is this drug, is this therapy helping? And that's also a primary interest of this study. So who can participate? Any children over 2 can participate, and so with any of the disorders that I mentioned, so XP, Fanconi anemia, Cockayne syndrome, CTD, are all involved in the study, or could be, and first degree relatives, meaning parents, siblings, or even children of a patient in some of these disorders. So, and then participation can be at any level of the study. So we have 3 parts. There's a survey, there's a blood sample collection, and a skin biopsy, different components. The only exclusions we have are pregnant women can't participate in the blood draw or the biopsy, and relatives younger than 18 can't provide a biopsy just for ethical concerns there. The survey, again, is health history information, just survey type questions about that. And then medical history, daily life, quality of life, sun exposure, those kind of things. And it should only take about 10 minutes or so to complete. The blood draw is less than 4 tablespoons. And the skin biopsy is just this 4 millimeter. It's just a really tiny punch they use. They use a tool that looks like this to just take the biopsy real quick. The pediatric dermatologist does this, who has experience in training in doing it. They numb it first, so there's no pain involved. They numb the skin first. And then there is one little stitch put in there and a Band-Aid, and it doesn't prevent you from anything. You can go swimming or anything you want after that. It's very low risk. Maybe bleeding or infection are pretty low, and maybe a tiny little scar from the biopsy. But it's really it's just a very small biopsy. And so within a few days, 7 days, it should be completely healed. And they do put a stitch, but it's a dissolving stitch, so there's no return or any real medical follow-up that you need to be concerned about for that. Okay, so what are we going to use the samples for? I should preface that we have maxed out our biopsy capacity for the study for this week. Unfortunately, we only had supplies. We didn't have everything organized with enough time to get all the supplies in, extra supplies for multiple biopsies. So we did have, thank you for all of those who did consent for biopsies. We do have all that we can do, but we can still take blood or answer the survey for anyone that's interested. So the tissue samples, we'll study DNA repair in them. We'll look for senescence, which is a type of cell aging, that phenotype that we look for in a cellular level, and also potentially eventually use these cell lines to be able to test therapeutics and the efficacy of them. The blood samples, again, will look for markers of aging or inflammation, antioxidants, and DNA repair capacity as well. And the survey will be able to use the survey information to match with some of the biochemical and blood samples and tissue sample readouts, and perhaps make some interesting conclusions from that interaction too. And the registry, as you're kind of hearing, this is really powerful to understand and characterize all of these disorders and how they relate to each other, how they're similar and how they're different, how therapies might work for one and not for another. These are all things that we would hope to be able to really delve into with these samples. So with the blood, we look at the PBMCs, which are the white cells that come out of the blood, and the plasma, which is when you spin it down, you get this clear layer of that we collect, and we can test for inflammation markers and aging markers within the plasma. And with the PBMCs, we can do test DNA repair, which is the NER assay. We can do genotyping, do an epigenetic clock, which is a way of looking at DNA and finding out how changes or modifications in the DNA give it more of an aged look or a younger look, and you can actually measure acceleration of aging in that. And of course, share with our collaborators as well. The skin biopsy, we take that tiny, tiny little punch, and we're able to even cut it in half and make the most out of it. Use it to the maximum we can. We freeze a portion of it, which we can then do some of this bigger genomic type analysis on it to really dive into some of those molecular questions about the disease and what causes it and what's different between the Cockayne and XP, or between the syndromes, but also between the family members too. And we can take that other half of it and culture the cells, and we can get 2 types of cells out of the skin biopsy. One's called a keratinocyte, and the other one is a fibroblast. The keratinocytes make the outer portion of your skin, the epidermis, and the fibroblasts are deeper in the dermis part of your skin. But together, we can use those to make 3D skin models in vitro, so we can actually test and grow skin in culture to do drug screening with. So with the DNA repair assay, one thing we've been really, we have gotten we worked with an XP family support group and collected a bunch of samples from that support group as well. And we were able to look at the XP patients and the parents and the siblings, and so we were able to compare both blood and fibroblasts from from all the participants. And you can see that our NER assay is very low. The patients all have very low DNA repair, whereas the parents have somewhat a spread of difference in their ability to repair DNA, and siblings as well. But that the siblings and parents were really no different. These are unaffected siblings, so they don't have the disease. And but you can still see that the patients have much lower DNA repair than either the parents or siblings. And then you can see that both in blood and fibroblasts, because our hope is that we could really move away from having to do this DNA testing in fibroblasts, because it's not that fun to have a skin biopsy compared to getting a blood draw. It's a little bit harder, a little bit more of a hassle. So being able to do this testing in blood samples is really nice, and being able to confirm that you basically get a similar result, whether you use blood or skin samples and blood is, you can get the answer right away. You can do the testing that week, whereas with fibroblasts, you have to grow up the cells for up to a month to get enough cells out of that tiny little biopsy to perform the experiment. So as I mentioned before, we can actually take those skin cells, those keratinocytes and those fibroblasts, and we can turn them into 3D skin models. So this is just what it looks like when we're culturing them in the lab. It's just in these tiny little inserts where you can actually grow human skin from these two skin cell types. And this is what the epidermis looks like when you can actually grow that, and it can grow and differentiate into, this is the outer surface, of your skin like that. That's what it looks like when you're looking at it under a microscope. So it's got all these different layers, and we can form all of those in cell culture, and then do a more relevant cell testing of therapeutics that are mimicking more of a skin barrier for testing. So data protection, all data will be de-identified. Your name or anything will never be associated with any of this. We would never sell any of this information, and it would never be linked back to you or your child. And there is no cost, so your insurance isn't billed or anything for this study. So participation, you can, if you haven't gotten a chance or you're on the fence about participation, or maybe we haven't had the capacity to have you participate at this event, you can always email and notify me or Christina Poole about participation. You would need to register as a patient at U of M just as part of the process for this, so that we can collect the samples, and consent, of course, is always essential for this. And both parents need to consent for a skin biopsy, because tissue is considered a little bit more invasive than a blood sample. So again, I'm Beth, and you can email me or Christina Boole if you're interested in participating too. We are also recruiting here, and we're trying to coordinate with Peter's group about doing the blood collections tomorrow here at the hotel. So does anyone have any questions about this? I'd be happy to answer them.